Hey guys, going to go through some cellular processes today. Putting together some of these tools that we see in the cellular toolbox. You'll see that a lot of these are just that, just combinations of these tools. Apparently my cat's going to be helping quite a bit. So we've talked about the secretory pathway, but we didn't take any notes on it before. This is during that part that I told you not to take notes. But you'll see that the secretory pathway and the endomembrane system, which is where all this takes place, it's just a combination of those cellular tools. But basically, the secretory pathway is the pathway we use to get proteins outside the cell or to get proteins embedded in the cell membrane, right? If we have a protein we want embedded in the cell membrane, part of it's in the cytoplasm and part of it's in the outside of the cell, it has to go through this secretory pathway. So we start in the nucleus with the DNA. And of course, we're going to use nucleic acid replication processes, right? Which is basically transcription in this case, because we're going to make RNA. So we're going to make an RNA copy of the DNA. That means we're going to unzip the DNA and copy the strand that's there by following those base pair rules. So that's how we make the mRNA with RNA polymerase. That mRNA can leave the nucleus. Of course, the DNA stays in the nucleus. So the RNA leaves the nucleus where it encounters a ribosome. And there are tons of ribosomes here in the cytoplasm. So it grabs it. As it starts to build it, the first few amino acids tell it, hey, this is a secreted protein, or hey, this is a membrane protein. They don't really tell it either of those things. What it really just says is stop translation, which is what this process is. When the ribosome uses an mRNA to make a protein, that's called translation. What happened in the nucleus with the DNA, that's called transcription. We'll talk more about those later. But so it'll say stop translation so that we can go to the ER and then we can complete translation there. So the ribosome stops translation until it encounters the ER, and then it will start to build the protein again, and it'll build it through the membrane if it's going to be a secreted protein. Here's a little video showing how that protein is made into the lumen of the ER. If it were a membrane protein it was making, it would build it here like this. So then that protein is going to be transported through the ER, kind of floating around in there, maybe get some adjustments, maybe get some editing, some changes happen to it. And then it's going to butt off a vesicle. And a vesicle is carried by motor proteins on cytoskeleton to the Golgi. So vesicles, motor proteins, these are some of our cellular tools, right? Here's a little video showing how that vesicle is hauled by a motor protein. It's walking on a microtubule, which is cytoskeleton. If this was a membrane protein, it would still be here. It would just be through this part of the membrane. If it's sticking through the membrane of the ER like this, when we butt off a vesicle, it'll be sticking through the membrane of the vesicle. That vesicle then is transported to the Golgi. And now if it's a membrane protein, it would be like this. But if it's a secreted protein, it's inside the Golgi. Again, some processing, some tweaks, some changes, some editing. Eventually, we'll butt a vesicle off of the Golgi. And then if it's going to be a secreted protein, we'll fuse that membrane of the vesicle, fuse it to the membrane of the cell, and that'll dump all of its contents out. It will also take that membrane protein and make it embedded in the cell membrane. If it's in the membrane of the vesicle, then it'll end up in the membrane of the cell like this. Notice how the split part, the Y part, is always in the blue. There was a vesicle here. And the tail part, this end, is always in the cytoplasm. Right? These areas, the ER and the Golgi, they are analogous to the outside. In fact, if you have a dye or a stain that you add into this solution with these cells, that stain will end up in the ER and then it will end up in the Golgi, because we are constantly butting off vesicles from the outside and fusing them to the ER and to the Golgi. And so we end up just with this whole cycle. It's like bubbles of membrane being transported, vesicles being transported from the membrane to the ER, to the Golgi, to the membrane, from the membrane to the ER, to the Golgi, to the membrane, from the membrane to the ER, to the Golgi, right? So we get this whole cycle going on. And sometimes we go from the Golgi to other places. This is how we make lysosomes and things like that. But their membrane is always being transported around, and there's always little samples of this space. Everything that I've colored blue here, except for the nucleus, that space is always being shared with the outside because of the way these vesicles are formed. So that's the secretory pathway. It's how we get proteins on the outside. And you should definitely know the order of these things. And you also should know where it's a protein, right? It's not a protein when we were back here. When we're back here at a ribosome, there's no protein yet, right? The ribosome builds proteins. We don't have a protein until we're at the ER. So the first place the protein even exists is in the ER because it's built into there. What we're seeing here, that's the ribosome. This is the mRNA. And down here, that's the protein being built into the lumen of the ER. Another process that cells do pretty regularly is apoptosis. This is programmed cell death. This is cells dying deliberately. And this is a big part of development. If you think about organisms that go through metamorphoses, like a frog, it has to lose that tail, but that tail is full of resources. It doesn't want to just chuck it and chop it off and let it go. It's going to use those molecules again in other cells as it grows to be a larger frog. Of course, when you were very, very small and still inside your mother, you had webbed fingers, you had a tail. Apoptosis took care of these things. Sometimes it fails and you end up with fingers still fused together. That's not uncommon. So apoptosis is programmed cell death. And what initiates is some signal usually from outside the cell, but it can come from inside the cell too. If there's something really wrong with the DNA, if there's major defects with the DNA, the cell can identify that and go, this cell should stop and let's just shut it down. And they will give themselves basically this signal saying we need to die. But a lot of times it comes from the outside. Like in the case of the web fingers or the tail, you're going to get a signal from other cells saying you're not the cells that we're keeping. So please put your affairs in order and be ready to die because it's going to happen. Now, when a cell gets this signal, it's going to do a few different things. It's going to open the lysosomes, which of course are filled with digestive enzymes. And so you're going to start digesting all of the inside of the cell. If the lysosomes bust open, all the proteins, the DNA, everything that's inside that cell is going to be broken down into monomers. And this is useful because we're going to recycle all of those components. We're going to use those again. So we can have nucleotides so we can make DNA in new cells. We can have, amino acids so we can make new proteins and things like that. So we're going to open the lysosomes and digest everything that way. It's also going to cause us to disassemble the cytoskeleton. And that's called blebbing. And basically the cell loses its shape. It just goes bloop, and it physically looks very different. It's kind of a nice little diagram here of a blebbed out cell. And then another thing they're going to do is they're going to label the membrane for phagocytosis. And that's basically getting eaten by a big white blood cell. So all of these little pieces, as these cells kind of shatter apart and bleb out, they're also labeled so that white blood cells will identify them and go, oh, hey, we need to digest this. And they will pick it up, fuse it with their own lysosomes, and then dump out the monomers so that other cells have those available. So basically, it's digest all the stuff on the inside in a very orderly fashion, disassemble the cytoskeleton so it's in small pieces, it'll fragment the cell membrane, and then label the membrane for digestion by other cells. And it's a very orderly way to kind of recycle all of the materials that were used in that cell. This is really common in plants and animals. If a plant cell gets an infection, they will sometimes tell all their neighbors, hey, I've got an infection, and it will make the neighboring cells. So like if you're looking at this little black spot down here, down at the bottom, some cells in here got infected. And so the cells out here set up a little wall. They basically made their cell walls really dense and packed them full of this waxy substance to try and prevent any of the infection, whatever the bacteria it is, try and prevent it from getting out of that area. And all the cells inside of there will receive the death signal so that they just die. So those cells die and make it so that there's not so much for the bacteria to thrive upon because we're not delivering it nutrients anymore. And we're going to wall it off so that it can't spread into other cells. And that's the hope. And so you can see this a lot of times in plants. You'll see these little spots. That's them fighting off an infection. It's tough for plants because every plant cell is essentially connected to other plant cells through those plasmodesmata, right? Through those tubes, through those continuous cytoplasm from one cell to the next. So we really have to wall that stuff off very quickly when there's an infection because otherwise things spread around very easily. Now we need to talk about cell transport, how we move things across the cell membrane. We've talked a bit about the membrane, but we need to make sure we're clear with the three main types of membrane transport. First of all, you need to make sure you understand diffusion, which is pretty straightforward. If you've encountered this before, if you've smelled, somebody cooking food, or you've added, Kool-Aid to water, you can see the colors spreads everywhere from a place where it's concentrated to a place where it's not. This is diffusion. It's this second law of thermodynamics. It's this tendency toward entropy. It's just things going to where there's less of those things until it's all about the same. Right? So when we're talking about a gradient, we're talking about a difference in concentration. So you can have a lot in one place and very little somewhere else. We are always going to have movement, right? The natural tendency of the universe is to go from where there's a lot to where there's less until it's all basically even. And that's what you're seeing in this little GIF here. So always from high concentration to low concentration. Sometimes we'll say down the concentration gradient, or we'll say along the concentration gradient. That just means like the easy way. And so to do it the other way, to go against the concentration gradient or up the concentration gradient, that's usually quite a bit harder, and it requires energy because that's not the way the universe wants to go. So when we got a membrane, like I said, there's 3 different ways things can move across that membrane. And the simplest one is called simple diffusion. This is hydrophobic molecules will go right through. Gases will go right through. Like our nonpolar gases like O2 and CO2 and nitrogen gas, which is basically everything we need to worry about. In fact, we don't even really need to worry about nitrogen. But all of those go through cell membranes without really any problem at all. So we're looking at things that don't have a problem with a hydrophobic barrier. Anything with a charge, anything with partial charges like polar molecules or hydrophilic molecules are not going to be able to make it through that boundary. I think we've covered that enough. So simple diffusion, hydrophobic molecules, and gases. And that's all you need to worry about. They're going to move along their concentration gradient from where there's more to where there's less. Some things would like to go down their concentration gradient through the membrane, but can't make it through that hydrophobic boundary. But if you put a protein channel there, then we facilitate that diffusion. We make that diffusion possible. And this doesn't take ATP on its own. Of course, we have to take ATP to build the protein, but once the protein is there, it doesn't take ATP to transport this, and things are just going to move along their concentration gradient. They're going to be free to move from where there's a lot to where there's little. And so facilitated means we have a protein channel involved, right? We've built a channel. That's all we mean by facilitated. It just means that there's a protein there that allows it to happen because it wouldn't be able to otherwise. Of course, we also have active transport. Active transport means you have to spend ATP. And we do this, again, when we're going against the concentration gradient. If you're going uphill, that's going to take energy because it doesn't want to go that way. It wants to go downhill, right? It wants to go from where there's a lot to where there's a little, and we want it to go the opposite direction. So that's always going to take energy. That's called active transport. That's the active part of it. It takes ATP. Couple of GIF GIFs here. You can see a couple of different styles. These are antiports and symports. And that's just a matter of whether they move together or whether they move in opposite directions as these proteins change shape. They don't always do either one of these things. Sometimes they're just pumping one thing. We talked about proton pumps with the hydrogen ions in the electron transport chain in mitochondria and photosynthesis. So those guys moved protons in one direction, and that was all they did. That's kind of simple active transport. They are going against the gradient, pushing protons where there was more of them. And so that's active transport, and it requires some sort of energy. In that case, it was energy from high energy electrons instead of ATP, but it has to come from somewhere. So typically it's ATP, but it isn't always. So active transport, like I said, is different from facilitated diffusion in 2 basic ways. One, we're going against the concentration gradient, and that's why the second thing, it requires some source of energy. Sometimes the source of energy is ATP directly. Sometimes the source of energy is, adding some other molecule or electrons. In the case of the electron transport chains we talked about, sometimes it is indirect, like we spin the ATP one place to do active transport somewhere else. And you'll see examples of that. So when you eat, you digest chains of proteins into amino acids. You digest chains of sugars like starch into individual monomers, into individual sugars, glucose and fructose and things like that. We're going to do active transport from your small intestine, from that lumen of your small intestine, which again is technically outside of you, right? That's not where your blood is. It's not where your cells are. That's still kind of outside. It's a fancy outside, but it's outside of you. And we want to bring those molecules in, those monomers. We want to bring in glucose, for example. It's a perfect example. Or amino acids. We want to bring those in. And we're going to use active transport to do that. Now, pause the video and think about that. Why would we use active transport to bring that stuff in, because there's obviously a lot more of it in your food, at least to begin with, than what you have in your blood. So why would we bother doing active transport? Pause the video and see if you guys can figure that out. So if you think about it, if we're not doing active transport, if we just have channels for glucose, here's the small intestine. Here's where your food is in the small intestine. You've got a cell. You got the wall of your small intestine, and then here's your blood and tissues, and this is you. That's your food. This is you. If you just have channels that allow glucose to come in, glucose will come in, definitely, because there's more glucose out there than there is in these cells. But that is only going to be true for a while. You're going to be taking this glucose and using it somewhere else. But at some point, these two are going to reach equilibrium. And you're going to stop bringing in glucose, or at least for every glucose you bring in, there's going to be one going back out because they're at equilibrium. We don't want equilibrium. We want every freaking glucose that we can get out of here. So we're going to use active transport to get all of it. That way you get all of the nutrients from your food instead of just half of them. They're valuable. Grab all you can get. Now, this is a thing that AP asks about a lot. They give you a lot of different examples, so I can't give you, every example. There's just too many of them. But I can give you an idea of how this works. It's secondary transport. So it's a way that we move a variety of things by only pumping one thing. The easiest example to give you is in plants. In the roots of plants, they are constantly pumping out hydrogen ions. And as they pump out hydrogen ions, they make a gradient. So there's a lot more hydrogen ions outside than inside the cells of the roots. So how this proton gradient works is kind of twofold. Anything that's positively charged is going to want to come in, because by pumping out all of these positively charged hydrogen ions, we've made it very negative inside the cell. We'll talk more about this later. This is a membrane potential. It's an electrical potential, meaning that it makes charges want to move. The way a battery works. Batteries make electrons want to move. They have a voltage. Cells have the same thing. Basically, all cells have this, but plant cells do it by pumping hydrogen ions out. So it makes it negative inside. So all I have to do is open a channel, and I'm doing active transport of potassium. It's going against its gradient, but it's not actually I'm not spending ATP right here. I just opened a channel, but anything that's positive is going to want to come in, because it's so positive outside and so negative inside. It's just charges repelling each other outside and charges attracting each other inside. Now they can also pick up nitrate, which is a negatively charged ion, and they do this with a co-transporter. This is just a channel that allows nitrate to come in, but nitrate doesn't want to come in because it's probably more inside because you're doing active transport of it, which you'll see here in a second. But the hydrogen ions want to come back in. And so what they have is a protein that lets the hydrogen ion come back in, but it only is allowed to come back in if it has a nitrate with it. And so when it has a nitrate with it, then they both come in. Hydrogen ions are like, let me in. They bind to that protein. And if a nitrate binds, then they can come in. And so that is driven by the concentration gradient of hydrogen ions. This is a lot like our ATP synthase, only instead of generating ATP because of the hydrogen ion gradient, we're using that energy instead to transport nitrate against its concentration gradient. So we can use that energy. We kind of like stored the energy in that gradient. We spent ATP back here with the proton pump, and that energy is kind of invested in that concentration gradient. So we can exploit that concentration gradient to bring in things against their concentration gradient. So there's a couple of different ways that we do secondary transport. One using the membrane potential, and one using a co-transporter. In every science class, they do this direct thing. You'll hear it all through college. If you major in science, you'll hear it through direct sources of X and indirect sources of X. The direct source of energy for the active transport of that potassium we were talking about, the direct source of energy is the voltage, is the membrane potential. Right? It's the difference in charge. The indirect source is the ATP, because we spent that ATP to make that concentration gradient, to make that electrical difference. The direct source of the transport of the nitrate is, again, this hydrogen ion concentration. So it's that movement, it's that gradient of hydrogen ions that powers that nitrate movement. So that's the direct source of energy. The indirect source, again, is that ATP used to generate the gradient in the first place. All right, so simple diffusion, real easy, gases and hydrophobic molecules. Facilitated diffusion, hydrophilic compounds, whether there's ions or sugars or whatever, if they're hydrophilic, if they have any kind of charge, they're going to need a channel. They're going to go along their concentration gradient. Active transport is going to require some source of energy. It's usually ATP, but not always. And it can get a little bit more complicated. So keep an eye out for that secondary transport. I want to talk about that electrical potential we just mentioned in that previous slide. If the outside of the cell has a lot more positive ions than the inside, and the inside has many more negative ions than the outside, then it's going to be positive outside the cell, negative inside the cell. So if I open a channel for, say, sodium, which is positively charged, right? A sodium ion has a +1 charge. If I open a sodium channel, regardless of what its concentration is like, it's going to want to come in because it's negative inside and positive outside, and the sodium is positively charged. So it's attracted to negative things. So it's going to come in. Does the concentration gradient affect this? Yeah, it does some. If I'm also pumping sodium in there and there's a lot more sodium inside than outside, then yeah, that's not going to probably move as much. It's going to be canceling that negative charge also. So I don't know how you'll generate that without doing the sodium thing anyway. So an electrical potential, of course, is just something that makes charges want to move. This is what you have in a battery. Electrons want to move from one end to the other, and you can put things in the way as it goes by. In the cell membrane, we get electrical potential by pumping positive ions outside of the cell. It's pretty much always positive ions being pumped out, usually hydrogen ions or potassium ions in plants, and in animals, it's usually sodium ions. And that electrical potential across the membrane, that electrical difference, is called membrane potential. Okay, so it's that electrical difference, negative inside the cell, positive outside the cell. Pretty standard for most living things. The membrane potential of most of your cells, and most animal cells, in fact, is about 70 millivolts. So it's not a huge voltage. And in humans, it's maintained, this should say, and in humans and animal cells, this is maintained by the sodium potassium pump. It's a pump, uses ATP to pump sodium outside the cell, and it pumps potassium inside the cell. It actually pumps 3 sodiums out and 2 potassiums in at the same time per 1 ATP. And now the cell also has channels for potassium that allows the potassium to leak back out. So potassium doesn't really want to go out because it's positive out there and negative inside, but we make its concentration gradient high enough that it does want to go back out. So you end up with more positive charges outside because all the sodium's out there, or almost all of it, and still a lot of the potassium is out there. There's still more potassium inside than outside, but there's still a lot of that potassium is leaking back out. So it ends up being a lot more positive outside than inside. This becomes really important when you're talking about neurons. This is how neurons and how muscles work. So let's take a little sidetrack here and look at how neurons work. Now, AP doesn't ask specifically about neurons anymore, but if you understand how they work, you can see that it's a lot of this active transport and channels kind of timed together that allows you to have very quick reactions to things. So first of all, neurons have polarized membranes. It's positive outside, negative inside. The membrane potential, again, is about negative 70 millivolts. That's the difference. That's basically the inside of the cell is about negative 70 millivolts compared to the outside, right? These have to be relative. It's maintained by the sodium-potassium pump, pumping the sodium out and potassium in, and again, letting potassium leak back out again. Not much sodium coming back in. So if sodium channels open, first of all, we're pumping a lot of sodium out there, so the sodium wants to come back in. If I open a channel, it wants to come back in because of its concentration gradient. But it also wants to come back in because of that membrane potential. It's very positive outside, and sodium is positive, so it wants to come back in to where it's negative to try and neutralize that charge and just to be with more negatives, right? It's an attraction thing. So sodium rushes in. We're talking milliseconds. If potassium channels open, there's still more potassium inside than out. So potassium will go out. Not quite as fast, but we'll say it rushes out. I'm not going to go all caps. But it does go out. So this is a situation before we want to send a signal. This is the resting potential, the resting situation for a neuron. Penguin asks an important question here. When is this pump running, the sodium-potassium pump? It is always running. Okay, so this whole time, as we go through all the rest of this, the sodium-potassium pump is always running, always trying to pump sodium out, always trying to pump potassium in. Now we're going to open some channels. It's going to mess with where things are, but the pump is always working. This was sent to me by a former student. It's a sodium-potassium, pump. How does a nerve impulse travel down these cells? Now, remember, some of these cells are very long, maybe over a meter. Resting potential is negative 70. We can depolarize it. So far what we've been talking about is this resting potential. It's polarized at negative 70 millivolts. Depolarizing means taking it back towards zero, okay, making it more neutral, getting rid of that membrane potential, making it go towards the positive. Repolarization means taking it back to that negative 70. Hyperpolarization means taking it lower, farther than negative 70 millivolts. If we go to negative 80 millivolts, that's hyperpolarization. That's more polarized than what it normally is. So these three terms, we're going to use them a lot, so you need to make sure we're good with that. Remember, resting's negative 70, and then everything else should make sense just by using prefixes. D means taking away that polarization, re means polarizing it again, and hyperpolarization means overpolarizing it. So some stimulus, usually from another neuron or something like that. Some information tells your brain, hey, I need to move something. There's something coming at my face. I need to dodge it, whatever. We send a signal to a neuron. That signal causes depolarization on the neuron. And right at the beginning of an axon. So real quick, a neuron looks kind of like this. It's got all these dendrites on it. It's got one long axon that then communicates to some other cells. This axon is where we are. Right, this is what sends the signal. So right here at the beginning of the axon, that's where we're making the decision to send this signal or not. And if you start the signal, it goes and you can't stop it. But this cell is receiving all kinds of input from other cells saying, hey, fire, or hey, don't fire. And that all adds up right here at the beginning of the axon. And the axon is going to be the place where we make the decision to fire. If that gets a depolarization at the axon above about negative 55, so it's at negative 70 resting. If it gets signals that make it go up above negative 55, I have voltage-gated sodium channels. They're going to open up. So if I go above negative 55 millivolts, channels open up and sodium rushes in. Think about what that does to our polarization, right? It's negative inside, it's positive outside, but a bunch of positives come rushing in. So that's going to kill our negative inside, right? So our membrane potential is now toast. It's gone. So sodium rushes in and it actually becomes positive on the inside. That makes a sodium channel right here, right next in line, that makes it open because it's voltage-gated sodium channels because it's going above negative 55. And so that channel opens and that brings in more sodium, which makes a channel right here open. And so we keep doing this, and this wave of opening sodium channels moves down the line because every time I open, I let in a bunch of sodium. It gets very positive inside, which causes these guys right, because that's going to spread from there. And that makes these guys right here open up because they're voltage gated. Because as that positive comes in, it changes the voltage on that membrane. Now, the potassium channels are also voltage gated, but they're on a delay. So the sodium channels open right away. And then the potassium channels go, oh, we got depolarized. And then they open. They wait a second, and then they open. It's not a second, it's milliseconds, but you get the point. So there's this wave of sodium channels opening, followed by a wave of potassium channels opening. So the sodium channels open, and that brings the membrane potential way up because it was at negative 70. And as we open sodium channels, sodium rushes in. So the membrane potential goes up. It makes it less negative inside because there's positives coming in. And it will actually swing up above positive. But then the sodium channels slam shut at about the same time that the potassium channels open. So now potassium starts leaking out. Sodium's still being pumped out by the pump, always running, right? And so as this potassium runs out and the sodium's being pumped out, it makes it more positive outside and more negative inside. We're resetting the membrane potential. We're putting it back to the negative 70 that it was at before. And this wave goes down the line. And you can see we're set back up, ready to go again. It looks just like it did before. And this wave is going to travel down the line. Now, what's actually physically moving? Nothing. Moving down the line? Nothing. Only stuff moving back and forth across the membrane. Sodium and potassium moving back and forth across the membrane. That's all that's happening. But it's like dominoes. There's no domino. When you push over a string of dominoes, no domino goes from one end to the other. This domino causes this domino, causes this domino, right? And it triggers the next one along the way. The thing that's cool about this is that the dominoes get set back up right afterwards. So it travels in a wave down the line, and it's extremely fast. So if I'm looking at one particular point on a neuron. So here's a piece of an axon, right? And the wave's going to travel down here, down this line. Sodium channels opening and then potassium channels opening. If I'm looking at one point right here, let's say I've got a little probe in here and I'm measuring the voltage. Here's what I'm going to see as that wave goes by. I'm resting before the wave gets there at -70. But as these channels right here open up and sodium rushes in, it's going to start getting positive right by me here. And so the membrane potential goes up. This is where that signal gets to me. And it starts going up. And so when it gets to here, right there, I open my sodium channels at that point. And so sodium comes rushing in. And so that makes the inside of the cell very positive, right? So this is sodium in. And then right there, the sodium channels close and potassium channels, remember, they're on a delay. And so now potassium is rushing out. Sodium is being pumped back out and no longer rushing in. And so this is potassium going out, and this is sodium being pumped out. This is channels because they're open, and this is pumps, pumping the sodium out. So we get down here and we go right past threshold. And we go right past threshold because those potassium channels are on such a delay, they let more potassium to leak out, like a little extra. And so finally down here, the potassium channels close. And that makes it so that we can go back to the way we started. We had sodium channels and potassium channels closed, and just the pump working. So this little bit right here, this is hyperpolarization. Right? We got more polarized than what our resting potential is. So we have depolarization right here. We have repolarization, and then we have hyperpolarization. And then we get back to where we were before. That hyperpolarization is really kind of cool. It's important because it makes it so that if I send another signal right away, this guy, even if I sent another signal to this neuron, it wouldn't do anything during that period of time. Right? It's too far away from threshold that would make it fire. Again, you don't need to worry too much about the details of this. Nobody's going to test you on neuron activity. I'm just trying to show you how we can get really complicated very quickly with channels and pumps. With just simple tools from that cellular toolbox, we can make very complex things happen. All we've got here is some channels and pumps, and we're able to make a neuron fire a signal in milliseconds so that, if somebody, throws a ball at your face, you can catch it, or at least dodge it. And it's just channels and pumps. So this is basically the way all neurons work. It's essentially they receive some signal, they decide whether or not they're going to fire, and if they do, they pass on a signal. If they don't, they don't. And you get input from your senses, right? Those are all neurons in your fingers of various kinds, and your eyes and your ears. All of these are different receptors that send action potentials. And that's what that wave of electrical charge is called. It's called an action potential. They send that signal to your brain or spinal cord in the case of reflexes, but to some part of your central nervous system where you fire a bunch of neurons that allow you to make a decision as to what to do about it. And then you send signals out to your body to do something about it. And those signals all work the same way with the sodium and the potassium moving across the membrane. Another good example you find in the nephron. Now, nephrons are like the fundamental unit, the functional unit of a kidney. You have millions of these in each kidney. You're going to do two basic things. You're going to filter the blood, basically squeeze all the small stuff through little holes. Big stuff stays in the blood. So I'm pumping blood. It's under high pressure in this artery here. Because like right back here is your aorta, which is right off of the left ventricle. So blood's coming in here. It comes into here. It gets squeezed through leaky tubes into this little pocket. So this pocket, this glomerulus, catches fluid from your blood. It's all the small stuff, not cells, not big proteins, but all the salts, sugars, amino acids, urea, all your wastes, all that stuff is squeezed into this yellowish tube here. And as it goes through this tube, then, if you look at this network of blood vessels that's surrounding that tube, it's actually much more complicated than what it's showing here. This is kind of a simple schematic diagram. We are pulling back the things we want. So we squeeze out everything that's small and we pick back up glucose and amino acids and some of the salts and things like that we want to keep. We pick those back up. until eventually they get into this collecting tubule, which then runs to eventually to the ureter, which goes to the bladder, and you pee it out. So you filter out everything in the blood that's small, and then you pick back the things you want. So you're filtering from the blood, and it's being powered by, that filtration is powered by the pressure in your blood from your heart. So we selectively reabsorb, which means we're going through a membrane, right? Selective means we have channels. It means we have pumps and channels, right? So we're going from inside of this tube. This is the proximal tubule, the loop of Henle, the distal tubule. You don't need to know any of those. We're going from that tube, though, and then putting it back into the blood. Only the things we want, though. The urea and the wastes, they're going to stay in there. We're not going to pump those out of there. So this is a really good question. Why do we reabsorb selectively? Why not just have, instead of squeezing everything out and then picking back what we want, that seems like we could just like dump out what we don't want and then we would be fine, right? But if you think about that for a minute, the things that we're going to select have to go through a membrane, which means they have to have a channel or a pump, which means they need a protein that is shaped in such a way to handle that compound, that molecule, or that ion. We don't know what we don't want in our blood. We only know what we do want in our blood. You might put all kinds of stuff in there. There's all kinds of compounds that you eat in foods, or when you take medicines, right? Your kidney would have to have a channel for every medicine you've ever taken to get rid of that. Your kidney would have a channel for every toxin that you could possibly encounter on Earth. There's no way it could be ready for that. So what we do is we dump out everything. We know what we want back. We want glucose, we want amino acids, and we want a few specific salts. And so we can just have channels and pumps for those things and bring all those back in and let everything else, regardless of what it is, let it go. And this is what happens, right? You're peeing out the ibuprofen that you take. As soon as your body starts absorbing it, you start peeing it out. And the same is true with any other medicine. Most of the compounds that your body can't do anything with in your blood, they end up going out in your urine. If they make it to your blood, they end up going out in your urine. We just reabsorb the things we know we want: glucose, amino acids, and some specific salts. As you go through that nephron, the proximal tubule, the loop of Henle, the distal tubule, again, you don't need to know the names of those, but each one of those absorbs different things, or reabsorbs different things. Think about how that could happen. Even pause the video here. How could you make it so that each section of that tube reabsorbs different things? Of course, the answer to that question is just by having the channels. Remember, channels and pumps are specific. So if you have channels and pumps for one compound, that's where you're going to move it. If you go into the next section, it has a different set of channels and pumps, so we absorb different compounds at those parts. The loop of Henle, that part that goes down, it's really just about reabsorption of water, and it does that using sodium and chlorine. So everything small. So to summarize the kidney, which again is a complicated set of stuff that's just channels and pumps. It's a very complicated process, but it's just channels and pumps. We squeeze out physically through leaky tubes using the pressure generated by the heart. Everything small goes out of those leaky tubes. They're basically capillaries, right? In that glomerulus. Squeeze out everything small. Proteins and cells, they stay in the blood. Everything else small gets squeezed out. What gets left in the blood vessel? Like I said, proteins and cells. What gets reabsorbed? Glucose, amino acids, everything useful, everything our body wants to keep. And what leaves in the urine? Well, that's everything else, but especially our waste products like urea. How about this question that the Jawa asks? Sodium ions are pumped because we pump the sodium back out. And the chloride ions, we just need a channel for. Why is that? Well, if you think about that membrane potential thing we were talking about, it's positive out there. If I open up a door for chlorine, it's going, right? This is the thing we do a lot. We do this in our lungs when we generate mucus in our lungs. We pump sodium, open a door for chlorine, and it'll follow. So sodium chloride moves right across the membrane, no problem. If you just pump the sodium, the chloride will go because of its charge, right? It's opposite charge. It's attracted to that positive environment you're making by pumping the sodium. Cystic fibrosis is a problem with your chloride channels, and you can't let the chloride go with it. It makes it so you don't transport as much water. When we move sodium and chloride together like this, we are almost always actually trying to move water. So you pump the sodium, chloride ions follow because of their charge, water follows because of osmosis, and we'll get to that in the next PowerPoint. One more thing about cellular transport we need to talk about is those vesicles. Just want to make sure you know these words, endocytosis and exocytosis, but it's basically this whole idea with the endomembrane system and vesicles. We can bring in a vesicle and it's just a little sample of the outside world, or we can fuse a vesicle and we can dump things outside. So it does require energy to bring these in or to fuse them. It's a complicated process with a lot of proteins. We're moving a lot more stuff than a channel or a pump would. But I think we've kind of covered that enough with the secretory pathway. It is interesting to note, though, that there are exovesicles where it buds off the membrane on the outside. And this is one of the newer things we're discovering in science, is that cells communicate with each other by budding off a vesicle, transporting it in the extracellular space, and fusing that to the neighboring cells. And the neighboring cells can know what's going on based on those. So it's, besides hormones and neurotransmitters and things like that, they can talk to their neighboring cells by making exovesicles. And we're seeing that some cancer cells do things differently with exovesicles. So it may be one of the treatments we use for cancer. It may be something we attack in a cancer treatment later on down the line. So anyway, that's a bunch of cellular processes. We still got to talk a lot about osmosis, and there's a couple other things we need to hit, but we're getting close to done with this cells unit. So hang in there. You're doing great.